So, you've been doing cell therapy a while, starting with melanoma, and I think that kind of around the time, maybe a little before CRI first funded you as an investigator in 2000, when I believe you were working with Phil Greenberg at Fred Hutch, right?
Yep. Yep.
So, now, through your CRI Chordoma Foundation CLIP grant, you're going after a type of rare cancer called a sarcoma, which doesn't respond as well to current immunotherapies, and in particular, focusing on a type of sarcoma called chordoma. So what motivated you to go after this type of sarcoma, and why don't current checkpoint immunotherapies work well in this type of cancer?
Okay, those are great questions, and I'm glad you're dating me back almost 20 years, I think. And I think that, let me just preface this by saying that cancer research supported our work at a time when practically very few other agencies and the NIH would step up to the plate. So, really grateful to Lloyd Ode and Gio and everybody else who has sort of enabled us to move forward in a time when this was a bit of a desert landscape, I guess you could say. I think that melanoma at that time, and probably still is, because 60% of people still are not cured from melanoma with immune checkpoint therapy, although that's been a major advance compared to even 5 years ago. No question about that. It is sort of a gateway cancer for immunotherapy and adoptive cell therapy in particular. Moving beyond adoptive cell therapy for melanoma to other solid tumors has always been a goal of our lab. And I'll point out that during this time, a lot of amazing postdocs and fellows and graduate students have contributed to this. One of these individuals, Seth Pollock, was working on sarcoma, identified mixed round cell liposarcoma as expressing broadly NY-ESO antigen. So we began targeting that, as well as synovial sarcoma. When I moved to MD Anderson, and Seth continues in that work, and he's just a wonderful colleague now. That's very rewarding to see, to continue in this field in sarcoma. So I just wanted to give him a shout out because he continues working in sarcoma cell-based therapies. When I moved to MD Anderson, one of the most unique aspects of this center was that it had individual clinics, even for rare tumors like synovial sarcoma, mixed round cell liposarcoma. Dr. Samaya and I worked on that project for a little while, targeting NY-ESO. But as I began to look at other hematologic and solid tumor malignancies, which were refractory to conventional therapy and even to immune checkpoint therapy, it turned out there were several rare tumors in which not only was immune checkpoint therapy not effective, but there were actually no good targets for T-cell based or even CAR-based therapies. Fortunately, again, being in the right place at the right time, we began an antigen discovery pipeline, which involved the identification of epitopes and peptides found on the MHC of tumor cells. And these methodologies allowed us to go where the money is, which is straight to the peptide being presented by these tumor cells that T cells recognize, to elute them off the MHC, run them through a tandem mass spectrometer, push it through a number of algorithms to filter those of the highest priority and the most immunogenic, not just for common tumors, but also rare tumors. And that took us to the path of a few different rare tumor types. Anthony Conley, Tony Conley, as some of you know, is a proponent and a clinician who sees a lot of patients with chordoma, had a prior relationship with the Chordoma Foundation, and we saw the opportunity to work in this area because of the CLIP award that Cancer Research Institute sponsored. And it so happened that some of these antigen targets, which we found to be immunogenic for common tumors, were also shared with rare tumors. And the only way we were able to make this connection was because there was already a database established in chordoma for RNA-seq expression. So what we did was cross-check that expression with our existing immunopeptidome database and found that there were several antigens and corresponding epitopes. which were potentially expressed in chordomas. Just getting back to your original question, one of your second questions, which is why, do conventional therapy or even immune checkpoint therapy has not been entirely successful for chordomas. I can't give you the entire explanation, but certainly there are not a lot of T cells infiltrating chordomas. And in addition to that, the environment, whether it's the osseous environment or whatever it is, niche that these tumors grow in, may be rich in suppressive factors, whether it's TGF-beta or MDSCs. And so there are other immune suppressive mechanisms which are preventing whatever T cells are there, whatever immune checkpoint blockade you're using, from having its full effect. And so immune checkpoint therapy works as long as there are T cells there. If they're not T cells, well, you really don't have a vehicle to take the brakes off the system. And so in a way, we're supplying those T cells. And I think to that point, we really need to do this in combination with either checkpoint therapies or other strategies to undermine the tumor immune evasive mechanisms. Sorry, let me stop there. That was a long answer to your questions. Yeah.
That's fine. So I get the sense that it starts with the tumors don't have many mutations, which makes them not that immunogenic to the immune system. The immune system doesn't come in, so then there's nothing for the checkpoints to work on. And so your approach then now with the cell therapy is to try to kind of, like you said, go to where the money is and find what are those targets on the tumor cells. And then you can kind of reverse engineer cells to create an immune response, I guess, from scratch against it. So could you I have two parts for this question. One, general, just could you tell us how this search is going? And then also as part of that, are these targets, you mentioned NY-ESO-1, which could be potentially shared between different patients.
Right.
Are the targets you're looking at now, are some of them shared between patients too, or are they more the unique neoantigens that would be different for each patient?
So neoantigens and neoepitopes certainly contribute to the immunogenicity of a tumor, and as everyone knows, may relate to tumor mutational burden as well as a correlation with response in patients receiving immune checkpoint blockade. However, I think equally important, or at least as important, are non-mutated tumor antigens. So these are shared tumor-associated antigens, probably the one that most people are familiar with, and certainly the CRI has played a major role in promoting and in the discovery of cancer testis antigens, things like NY-ESO or PRAME or MAGE antigens. These are non-mutated tumor-associated antigens that are selectively expressed in tumor cells and also germinal tissues, but that makes them relatively safe to target. There are other tumor-associated antigens which are overexpressed in tumors and only minimally expressed in some tissues. One clear, obvious example are the melanocyte-associated antigens like MART1 and GP100. We can still treat and cure patients without serious toxicities, giving T cells to recognize these targets. And then there are those which are associated with some functional property that enables the tumor to outgrow its normal tissue counterparts or gives it some tumorigenic or metastatic advantage. These are overexpressed in some tumors. And our immunopeptidome profiling is most, so the immunopeptidome profiling that we do is been most adaptable to the discovery of tumor-associated non-mutated tumor antigens. And
just overexpressed pathways then?
Yeah, so cancer testis antigens, overexpressed tumor-associated targets. As it turns out, a number of antigens that are associated with immune resistance and tumorigenicity. Now, these are not mutated. I think ideally, one would have to demonstrate the immunogenicity of this to prove that these tumor-associated antigens present epitope with sufficient density on the surface that they can elicit a T cell response. And that's part of our engine discovery pipeline, is to prove, in fact, that these epitopes not only are there on the surface, but they're also immunogenic. And I think that empiric validation of these immunogenic epitopes is critical for tumor antigen discovery, vaccine strategy, cell therapy, obviously, and even antigen-specific immune monitoring. I think one of the obstacles in the field, I guess, or one of the challenges in the field is that people taking a RNA-seq database or some expression analysis, or even focusing on mutated regions of potential neoantigens, pass this through a in silico sort of algorithm to predict potential epitopes. And myself, maybe other people, believe that while that can be productive, there is a very, very high fraction of those predicted epitopes which are not at all immunogenic, which may not be presented at all. And I think that needs proper validation. The pipeline that we've been using and that has been applied to the treatment of chordoma and antigen-specific T-cell-based therapies is the direct result of a mass spec identification of eluded peptides followed by empiric validation of their immunogenicity. So we're fairly confident that these epitopes can elicit a T-cell response and hopefully eradicate tumor.
Are you looking at primarily MHC1 associated epitopes, or both?
Good question. Yes, I mean, we've had a long history of looking at both CD8 and CD4 T-cell responses. One of the first publications quite a few years ago demonstrated that adoptive transfer of CD4 T cells targeting NY-ESO could cure a patient with metastatic melanoma. And since then, we've continued to try and find methods to engender CD4 T cell response. However, the most, I guess, proximal and productive approach right now has been to elude class 1 restricted epitopes as a means to generate CD8 T cells. We also start looking at class 2 restricted epitopes, as many other labs have been doing. These are a little bit more difficult to nail down in terms of their immunogenicity, but I think we have the tools and infrastructure to do that. So I think the response to your question really is certainly no one T cell type can go at this alone. We certainly would like to engender a CD4 helper response. As it turns out, there may be ways to circumvent the requirements for CD4 T cells, not just through exogenous administration of cytokines and so forth. As an example, we've been developing the use of helper-independent CD8 T cells, actually since 2005, and have moved this into the clinic and made this more of a streamlined workflow in which we can routinely generate CD8 T cells that are early central memory type and make their own IL-2, proliferate long-term, and demonstrate that after adoptive transfer, these central memory type T cells can persist without the requirement for lymphodepletion or high-dose IL-2. So there are ways to address that issue. That's what we call epigenetic engineering, but there are obviously many strategies by many investigators, most of which actually that I know are supported by CRI, who have genetically engineered methods to give T cells, or CD8 T cells in particular, a durable, long-lasting function.
Great. And so a lot of the challenges that you've been discussing so far aren't necessarily unique to sarcoma, in that kind of once you validate this method and once the technology is proofed and up and running, it could presumably be used for other types of cancers too, right?
Yeah, I think, so it's interesting, it kind of goes both ways. You know, when we first identified the epitopes and antigens, they were in common solid tumors, and it turned out by cross-checking that RNA-seq, we could use them in chordomas and other rare tumors. And if that's a proof of principle for the ability and the feasibility and the effectiveness of chordoma reactive antigen-specific T cells to work, that antigen is also shared across many different solid tumor types. You actually have an entree into the ability of using the same target for which you've already de-risked it in a way for chordoma, and use it to treat patients who express a comparable antigen and restricting allele. I think in a way, I guess it is a gateway to solid tumor therapy. Using rare tumors such as chordoma as a strategy or as a clinical unmet need to propel the clinical trial and to initiate collaborations is a fantastic mechanism for getting the entire antigen-specific T-cell based strategies into the clinical arena.
Great. And real quick before, I just have two last quick questions. But first, I just wanted to clarify, you're not using a strictly TIL approach, right? You are actually engineering the TCRs into. Okay.
Thank you, Arthur. I mean, that's actually, just to make that point very clear, I mean, there are different modalities. One you can engineer with a CAR T or TCR-T, TIL, which requires tumor. But what we've been using is straight from the peripheral blood. And the reasons for that are that you have a broader T-cell receptor repertoire to choose from. It's obviously, you don't require tumor accessibility, and you don't require a tumor that has infiltrating lymphocytes. The difficulty, which has been the result of years of development of enabling technologies, is that you have to isolate those very rare tumor-reactive antigen-specific T cells, and we were able to do so successfully. But it required about 15 years of development, and so we're fairly confident.
And expanding that pool to a dose size would be its own challenge.
Exactly, and then we can expand that actually, with uniformity to billions of cells, and that allowed us to examine both the reasons for success and failure in a more rigorous fashion, and also, of course, to be more flexible and agile on how we move this beyond melanoma.
Awesome. So you briefly alluded to it earlier, but I want to talk about combinations now, because even, as you're with these genetically engineered cell approaches, you can, and yours is relatively straightforward, but I know that there's also other efforts where they can engineer the cells further to give them more properties, whether they're resistant to PD-1 or whatnot. But it sounds like, or let me ask you, do you think that many of these cell therapies will still need to be used in combination moving forward to kind of get their maximum benefit?
So I think that the engineering, the synthetic biology really has moved the field into areas that we could only have imagined a few years ago, and are actually being put into practice. I think that definitely the ETC, as we call them, the endogenous T cells, or these natural occurring T cells that we isolate, have been in our lab and in other labs engineered to enhance their function. So I think it's a two-way sort of information synergy in which there are properties of natural occurring T cells, which people are trying to emulate in the engineered T cells, and there's engineering that's going on there that we can, in parallel, enhance the function of these transferred antigen-specific T cells. And this is the case in point is because we already have a T cell receptor that we know works well and has mechanisms for feedback and upregulation and downregulation in a natural process that allows these cells to persist. We believe that's important, expression and also downregulation. But also in a parallel path by either knocking out PD-1 or augmenting other pathways that enhance their durability, anti-apoptotic potential, for example, or even stealthiness can be important in getting these beyond sort of a boutique type approach to a more off-the-shelf streamlined approach. So that's intrinsic to the T cell, things you can do intrinsically. But I think in combination, we've used this process of a uniform population of a well-defined T cell, of clear specificity, clearly defined phenotype, as a sort of a transferable cellular biomarker. And I say this at every talk at the end, I don't know, maybe I'll try and brand it at some point. But basically, CRI can call it whatever it likes. But I think really anyone looking at this can say, hey, we've got a bolus of cells we're putting in there. We know it's tumor reactive and traffics and goes anywhere. But we can interrogate that cell during the process or in combination with all these extrinsic immunomodulatory modalities, whether it's a vaccine, whether it's radiation, whether it's a checkpoint inhibitor, whether it's an agonist, and so on. Just pull these cells back out, and now you've got a way to examine a population that is likely to be tumor reactive and is the one you want to influence when you add these combination therapies. So it might be one way to sort of examine with some rigor this, embarrassment of riches, I guess, of immune checkpoints and agonists that are now clinically available.
And I guess by being able to do that, by being able to look at the cells, you infuse the cells, you do some other type of treatment, whether it's checkpoint or something, then you can pull them out and look at them. You could almost kind of use that to study exhaustion or any other kind of differentiation program, because you can't, I mean, it would be hard to do that. You wouldn't be able to do that with the body's normal cells because you wouldn't be able to tell where they were when the treatment was given. But with these, you could kind of backtrack the whole process.
That's great. Exactly right. I'm going to write this down right now for an oral one. But basically, what you're describing is exactly the sort of thing that we're addressing because of all the tools available now, single cell RNA sequencing, multi-omics, ATAC-seq, and so on. We're doing this both in vitro because there are ways to simulate exhaustion of T cells in vitro with these antigen-specific T cells. And as you're saying, there are ways to interrogate them after adoptive transfer, either at the tumor site or peripherally, to say, do these correlate with response to mitigate against exhaustion or promote expansion proliferation in the right place at the right time?
Because essentially, other than the TCR, they are in every other way, their biology would be identical to a normal T cell.
Yeah. And when you think about it, for example, going way back, patients, in medical school, I learned that patients who develop infectious mononucleosis, you have just a few epitopes, a few T cells that expand 10,000, 100,000 fold, and your spleen gets super enlarged and lymph nodes gets enlarged. There's a potential for these T cells to expand to much greater levels than what we see as potential limits, even ex vivo. So I think the potential there for these T cells to work in even more robust fashion is present. We just have to figure out a way to unlock that and to prevent the tumor from suppressing that mechanism.
Now, I guess, just to be clear, with viruses, there's fewer antigens, so the immune system, kind of the immunodominance phenomenon where since they only have a few targets, all of them are going to target the same thing. But with a tumor, it's a lot different because there's so many targets that kind of the immune response gets diluted a little bit. But with your approach, because they would all be targeting the same thing, presumably you would amplify just that immune response you want.
Yes. So, and I think that's important to know, certainly from a scientific standpoint, to track and understand how a tumor responds to a high pressure focused response. But what's also important, of course, is that the tumor is highly heterogeneous. You want to be able to propagate that response to other specificity, to a certain multivalency. We demonstrated in a number of papers that antigen spreading is important. That might be facilitated by, lowering the threshold of activation of endogenous tumor T cells, which is why you might need to preserve that population of endogenous tumor T cells that are already present while you give a very selective antigen-specific response. So no question, that one T cell specific or multiple even specific T cells that we give to patients may not be enough. But our studies and others have shown that if you can induce antigen spreading, it could potentially be enough. You just need other strategies to help that along.
Trigger, get the immune system to do it. It's not that it was designed against cancer, I know, but to kind of kickstart the whole process and then let it do its thing. So I just got one more question before I'll let you go. You talked about it a little bit when we first got on, but I was hoping maybe you could kind of, just again, looking back, how far the field has come over the last 20 years. If you could just kind of share some of your thoughts about this journey, and in particular, like you mentioned, the role that CRI has played in advancing the field of immunotherapy.
When CRI started funding our work, you could fill my office, which is maybe 10 by 20 square feet, with all the audience members attending that talk. And I think that at that time, understandably, immune checkpoint therapy had not been put into clinical practice. We had a few scanned antigens. I look back at the work that other people have done. Terry Boone at the Ludwig Institute, Lloyd Ode, obviously, at Cancer Research Institute, my mentor, Phil Greenberg, and others. And you think about how you go back to first principles to understand the basis for what we're doing now. So a lesson I hope that people will take to heart is that please go back to first principles and understand the T-cell biology, what the mechanisms are. Don't go randomly over-engineering or over-incrementally devising new protocols unless you understand some of these first principles. Not that those studies aren't very, very important also, but those are the edges of the battle. But to get to where the strategies are going to arise from really involves some additional rereading of even past literature and appreciation of all the basic understandings of T-cell biology and the ability to be flexible enough to understand that those teachings may not always be accurate, always questioning that. The one last thing I will have to say in this arena is that as I was going through the process of moving from bench to bedside, I know it's a cliche, but it's unquestionably for cell therapy, one of the most rapid transitions that one can imagine from benchtop to bedside. I was also training graduate students and postdocs and physician scientists. And this is something that I learned through CRI and other funding agencies. This is critical. We have to support these individuals at a very early stage and to provide them the freedom to free associate, to try things that happen purely from serendipity. And I can't explain enough how important that is, especially to current funding climate, the ability to perform or pursue a vision or goal which may be completely imaginary, grounded in science, can lead to real change.
Very well said. Yeah, it was the part you mentioned about first principles. It's interesting because clinically immunotherapy has taken such strides over the last decade, and obviously we've learned a lot more. But it's been really, the way I see it, it's been we've gained a lot more understanding about some very narrow areas, and they've propelled us forward so far. And now that the cures are so much better, or the probability of cures is so much better for several types of cancers. But then, even like we were talking about earlier with the CD4 T cells, it's like, oh yeah, we don't really know what half of them do, but they're important. Right. So for me, I see it as a positive that there's so many more other avenues to explore. And once we do fill in more pieces of the puzzles, it's going to make sense why, nowadays it's so CD8 focused in a lot of ways. Because like you said, I mean, the most important thing is to save patients' lives. And right now, you can't always afford to, wait until all of it's figured out. We got to do the best of what we can now. So it's incredible how far it's taken us. But yeah, it's always funny to think back, and I think the field is good about knowing how much it doesn't know, but still so much to figure out about the immune system. It's incredible. But,
it occurred to me in a way that CRI has supported our lab at every critical stage so far. And it's hard to remember that until you step back from it and say, hey, listen, when we were doing those first trials, how was that possible? Unfortunately, we had no NIH funding for some of these seminal studies. Even the first trial in combination with CTLA-4, that was a CRI-funded trial. It was the first time that had been done prospectively in patients. And even getting the reagent for us to do that combination was really through the efforts of the Cancer Research Institute. Even putting us together, with pharma that allowed us to use or get approval for the use of IPI before IPI was approved is an unimaginable sort of barrier to overcome in any other way at the time. And now, for example, just by chance, happened to be working in chordoma, and this next, step beyond melanoma to rare tumors and how that might influence the next generation of researchers or that area of clinical discovery was sort of catalyzed by this chordoma grant.